Okay, we should get started now. Welcome back, everybody. Our next speaker is Professor Jennifer Ross from Syracuse University, and she will be talking about self-organization and self-propulsion of active biological building blocks. And she is very much willing to take questions in the middle of the talk, so you can either unmute yourself or type in the chat box or raise your hand, whatever option suits you find. So, Professor Ross.
Great, thank you so much. I really appreciate the invitation. This has been a really cool talk series, and I'm really excited to be a part of it. And it's great to see all the students and everybody who are, I can't see your faces obviously, but I can read your names. So I'm going to talk to you today about two recent works that we've had out published and with a little bit of new stuff added in, but you can find most of these things available. We're going to start with a quiz because I am an educator like many of you, and so I like to see where people are at. So I'm showing you these 4 movies to start, and if you could use the chat box to we're going to vote on each movie. So on the first movie, what you're going to vote on for actually all the movies is whether or not you think that this movie was taken inside of a real living cell, so a cell that's alive and crawling around, or if you think it was something that we recreated in the lab through an in vitro reconstitution. So let's do the first movie as a warm-up. So vote in the chat if you think this is from a live cell, or if you can just write the word live cell, or if you think it's from something we recreated, so you can just write recreation. So go ahead and add to the chat. Just this first movie. This is how you do interactive talks in Zoom era. So if you don't put anything in the chat, okay, we got some about 50-50 so far. Okay, recreations, lots of reconstructed, few live cells. Recreations is winning out. Okay. So there's 50 of you, but I think about 10 have answered. So I think that's a pretty good percentage. Let's move to the second movie. So this will be movie 2. So I'll write movie 2 so we can keep track. So now for movie 2, vote if you think this is in a live cell or recreated. And I'll just be clear that this is actually the same movie in 2 colors, red and green, and a red-green overlay. So if you think it's in the live cell, you can write live cell, or if you think it's recreated in the laboratory, you can write recreated. Okay. Okay, we've got a little bit more recreated on that one. Okay, so then we're gonna do movies 3 and 4together. And so let's do a vote on these 2. So do you think they're in a live cell, or you think it's something we recreated in the lab? Okay, so that one we've got a little bit more live cell, some recreated, a little more live cell. So with this quiz, it's a self quiz, so we'll go to the answers in the next slide. So if you're ready, done voting, we're gonna go to the answers on the next slide, except for, my slide is not advancing, here we go. So in the first movie is actually a live cell, so you can just judge for yourself how you did on this quiz. Obviously, no points will actually be awarded to your grade because there's no grade for this, but what it's showing you though is that I think a lot of times that when we're looking at these movies, they do there's certain intuitions that you have because there's certain things you're used to seeing when you see something that's alive. And in particular, we can also add into, what does it mean to be alive, right? So when you say something's in a live cell, there are certain characteristics that it has. So what are those characteristics? So if you want to add into the chat some of the characteristics, or you can unmute and speak up and say why, in particular, these last 2, people tend to think that they're often in live cells, but they're not, they're reconstituted. So why do they look like they're alive? So can move. Yeah, so you've got some nice dynamics going on. Does anybody else want to say why those look alive? They have fluctuations. Yep. And let's do one more. If one more person could say what makes something look alive? It seems randomly ordered. Yeah, but see, that's a little bit odd, right? You said ordered, which implies not random. And then you also said, you also added random as your modifier for that word. This is something that's very unique to biological systems. There's a clear order to them, but they do seem to have a lot of fluctuations and a lot of stochasticity and randomness to them. And so I'm going to tell you two stories today that are both talking about organization and including some fluctuations. And so I think that biological systems have so many really cool properties, and that's why we study them, because one, I think we want to learn new physics from biology. We also want to understand how the biology works, just for understanding biology's sake. And so today we're going to talk about the first tale I'm going to tell you about is microtubule spindle self-organization, and just some microtubule organization type of stuff that we've done recently. It was published in Soft Matter. And the second one is some new stuff I'm really excited about in the lab on enzymes and how they can actually be self-propulsive. All right, so let's get started on one of these talks. So the first one is on microtubule self-organization, and this was work done by mainly by two people in the lab, Bianca Edozi, who was an undergrad in the lab. She's now a graduate student at the University of Chicago, and Suman Sahu, who's one of my grad students who moved with me from UMass Amherst to Syracuse University. And I have some additional follow-up new stuff that Suman has done since then, which is currently under review at Physical Review E. So I think that biology is really, really amazing. And if you think about it, what's really cool about biology is that it has constructed things. It is able to build things like humans. And we're like a machine, right? We're kind of like, I sometimes think of humans as being like a meat robot. We're able to move, we're able to do all these things. We also have all these other cool attributes, like if you cut your skin, it'll heal itself. That's certainly something that normal, robots don't do. Certainly normal materials don't do that. But that's because that we're large machines that are actually created out of smaller machines. And so if you think about inside of the human body, there are organs. I'm using the brain as an example, but you could use the heart, the liver, your stomach, your intestines, any of these things. These are smaller machines that are coordinated within your body machine to do a function, right? And then if you go even further down, these machines are made of tissues and cells, and the cell you could think of as also being a smaller size machine. And then if you go even further down, what's really cool is all the way down, this is now the micron level, those kind of filaments that you're seeing in there are also machines. The microtubules, they can grow, they can push, they can fall apart, they can pull. And so they're able to produce work. They're active systems. Every single one of these layers is an active system, meaning that it uses energy to perform its function. But we don't really understand how this does this. And then even down, one level down from here, that microtubule is made out of tubulin dimers. And the tubulin dimers, you've probably heard a lot about them over the last week, given the speakers. They are also machines. These are enzymes. And now we're on the nano scale. But at every level, where you have an energy using subunit that is able to use that energy to perform work. And the way it does that is not random. It's somewhat random, right? Because there's a lot of fluctuations, especially when you're talking about in water. You've got a lot of Brownian motion and things like this, but it's also rectified. It's often in the microtubule, right? It's all pointing in one direction. It's broken symmetry, and that's how you're able to get work out of it. So we're going to talk about all of these concepts today in these two different talks, these two little tales that I'm going to tell you about. And so obviously when biology can do all these cool things, we really want to know how they can do that, because we want to learn new physics from this. And ultimately what I'd love to do is elucidate those fundamental principles to use them in future material systems or other kinds of systems that, can do really cool, amazing things. All right, so for our first tale, we're going to actually use the microtubules that I was just showing you. And I'm not going to do the background of microtubules because I know many of you have seen talks already on microtubules. But we're going to start off with, the way we're going to do the experiment is we're going to start off with tubulin dimers and with or without crosslinkers. And the amount of crosslinkers and the amount of tubulin dimers are going to be our control parameters. So what we're putting in here right at the beginning is the control parameter for this experiment. And then over time, these tubulin dimers are going to form microtubules, which will be cross-linked by these crosslinkers. And MAP65 is a plant microtubule associated protein. It's similar to PRC1, which you may have heard of. It's also similar to ACE1, which is from yeast, but it's basically an anti-parallel crosslinker between microtubules. Over time, these not only nucleate microtubules that then bundle, but it nucleates the whole bundle. And then ultimately, and I'm going to give away the punchline here, if you have enough crosslinkers, you will actually get these tactoid shape or football shaped, I guess I should say, be clear, rugby ball shaped create bundles of microtubules that are able to have this very particular shape because they're created out of subunits that are longer than they are wide, which are the microtubules. All of these experiments are also going to include a crowding agent. In this case, we're using methylcellulose, and I can tell you more about what happens with different crowding agents if you are interested in that. Just to be clear, also, we're going to be imaging these on our fluorescence microscope. We're going to be using total internal reflection fluorescence microscopy. The crowding agents are going to allow everything to kind of be smushed down to the surface so that we're able to see whatever the patterns are right on the surface. And so this is something that we use a lot in the lab. You've probably seen it before. It creates an evanescent field up about 200 nanometers up from the surface. So that's the area that we're able to image. So I told you that one of the control parameters is the amount of tubulin that we put in, and that's because the amount of tubulin that you put in controls the length of the microtubules. So in fact, what we believe, and we have a lot of evidence for now, is that the filament length actually is a control parameter for some of these patterns. We can control the length by just changing the amount of free tubulin, but there's another catch, which is that we're going to inhibit dynamic instability. So microtubules have the ability to both grow and shrink. And in fact, that's what the enzyme is used for, is falling apart or self-emulation. Again, another one of these biological attributes that biology can do, but we can't design yet. If you inhibit dynamic instability by putting in a stabilizer, in this case, it's GMP-CBP, which is a non-hydrolyzable analog of the chemical fuel GTP, you'll end up getting that they only grow and they don't really shrink. And when you do that, the length is now controlled by the nucleation and growth processes. So there's the initial nucleation. If you have more tubulin, you would nucleate a lot of seeds, and then they kind of all start growing at the same time, and nobody falls apart. So they all kind of grow together and they all end up around the same length. So we've quantified that here, and you can see as you add more and more of the tubulin, you get shorter and shorter filaments, and you get more and more of those shorter filaments. If you just do all of this in our normal way with GTP and then post-stabilize with something like Taxol, you end up getting a very wide distribution, and it's not very kind of set contour length, right? And so then that doesn't allow us to test the length as one of the control parameters. So when I said that tubulin was one of the control parameters, the concentration of tubulin, it's because it controls the length in these experiments. All right, so what does it look like in the microscope? So here's an example where we have 13.6 micromolar tubulin, which is a contour length of about 6 microns. And again, we have our methylcellulose helping to push everything onto the surface with depletion interactions. If we don't have any crosslinkers, what we see is that they start forming, and locally they'll align, but globally they're not aligned at all. And so we can actually test this with a variety of different tubulin concentrations, which again control the contour length. And what you see is the kind of a similar pattern globally, but the local domains are now being controlled by that length of the filaments. And so the pattern that you see, if you were to kind of take the pattern from the smallest filaments and then zoom in, it would look exactly like the ones from the longer filaments. It's just that the scale of those, or the domains that you're able to get, become larger and scale with the length. And interestingly, they don't scale with the length like length squared. They actually scale as length to the 1/3, which is surprising. We can look at these domains so we can create local areas where the microtubules are all pointing in the same direction. If you're interested in how we do that, I certainly can tell you more about that. When we do that, we can then look at the domain area. We can also take those images and do spatial correlation. And what we see is that indeed the length of the filaments or the concentration of the tubulin really does control that correlation length, and it also controls the domain sizes that we're able to see. And now you can see from the bar chart of the correlation lengths that that correlation length actually increases with the length of the contour length. Now we were kind of interested in this, but this seems clear. But to do these experiments is difficult and hard and takes a lot. And so my student Suman, who's probably also could have easily been a brilliant theorist, but instead decided he was interested in experiments. He took it upon himself to try to do some simulations of this using this lovely software called Cytosim from Francois Nedeleg. And he was able to, kind of in a 2D growth, you can see we start with little microtubules. So we don't allow nucleation. We actually start with seeds, but we allow them to grow. We control the total amount, so no shrinkage, only growth. We control the total amount of available polymers, so they all grow to the same contour length. And when he does this, he's actually able to recapitulate a lot of what we see and then perform the same domain analysis on this system. And when he does that, you can see this is just a snapshot of several different contour lengths that he can now program in. And you see, you get the same kind of scaling that you got before with the experiment. And so this was exciting. So we did the same analysis. And then again, you can see that truly now that we can test any number of contour lengths, you can really see that the contour length or the correlation length that you get in the system, the contour length is really controlling that domain size, and it's really controlling the contour length. So we're very excited about this because it kind of showed that we were able to recapitulate that in our simulation, implying that yeah.
So are the microtubules growing from the nuclei radially outward, or after growth they're arranging like that?
Yeah, that's a really good question. I mean, when we're looking at the movies, it looks like they're initially kind of random, and then they grow and they bump into each other, and that limits the domain. So it really seems like kind of they don't rotate very much. So they don't have a lot of rotational mobility even at the very beginning, which allows them to create these domains. Let me just ask you a question. Are you talking about in the experiment or in the model?
No, in the experiment. The model, of course, you did that.
Yeah.
I mean, you designed it that way, the growth from the middle.
Right. What do you mean growth from the middle? They do grow from both sides of the microtubule.
No, so I thought when you have the nuclei and then from the nuclei you try to grow, I mean, you have a nuclei at a point and from there you grow radially outward. Is that right? In the simulation?
Grow, I mean, let's look at this again. What do you mean by grow outward? They each one of those individual seeds. So let me pause this. At the beginning, each one of these individual seeds, and this is colored based on its orientation locally, right? Then each one is able to grow from both ends. And then they're just bumping into each other with steric hindrance, and that's what causes the alignment. Is that answering your question?
Okay, yeah. I got it.
Okay.
Yeah.
Great. Okay, so all right, so now we can start adding those crosslinkers in. And when we do that, what we see is a collapsing of that local alignment into bundles. And in fact, it's not like individual microtubules nucleate all over and then they collapse into each other. They actually nucleate as bundles, which is what I showed you in that initial cartoon. And what was really exciting to us was seeing that actual shape here. So just showing you one example here with my cursor. You can see several of them. But the fact that they were able to create this spindle shape, much like the mitotic spindle, was very exciting to us. especially because MAP65, again, its analog of PRC1 has been shown to be really important in the mitotic spindle shape development as in real cells, in live cells. And so if we just go back and look, so we did, as good physicists, we did a whole phase diagram, and you can see at the top here with very low, so this isn't zero crosslinker, but very low crosslinker, it looks pretty similar. Maybe they're starting to condense into some of these tactoids. And as you increase the amount of crosslinker, you get more and more of these tactoid shapes. So if you're, again, and when I say percent, this is percent binding. So we use the KD value for the binding to microtubules to estimate how many tubulin dimers would have, or what percentage of tubulin dimers would have a crosslinker on it. So if you're at 10%, then 1 in 10 tubulin dimers will have a crosslinker. If you're at 3%, then 3 out of 100 will have a crosslinker. And so so even at 1.5%, you're already starting to see that condensation into these bundles, but maybe still some disorganization in the background. You can see further condensation when you have 3%, and then at 10%, you really get these individuals like spindle-like things. They're very well separated from one another. And I should say that this also depends on the contour length, which is kind of interesting because as you have shorter contour lengths, you have actually more filaments, right? Because remember, we nucleated from more tubulin to get shorter filaments, but in actuality, you need fewer of the crosslinkers in order to drive that condensation with short ones, because the short ones kind of use up that nucleate and grow into the bundles easier. But so you can see with both the 6 micron and the 2 micron that you're able to get those nice individual tactoids, and they don't all perfectly form. Sometimes you'll get 2 that are kind of stuck together like this. That often comes when they've nucleated near each other. Interestingly, they don't merge, right? They're not like liquidy and merging the way that actual mitotic spindles are. We'll talk more about that in a second. So our first thing that we wanted to check is just make sure, is this really being driven by the crosslinkers? And of course, the first hint is that you don't have crosslinkers and you don't get this. And it seems to be dependent on the crosslinkers. But in addition, we can use a GFP labeled MAP65. And we can look at that in the green channel, which is in the middle. We can look at the microtubules in the magenta channel, and we can overlay them and really see that, yes, everywhere where you have the microtubules, you really have the crosslinkers present. And that's what's driving this condensation.
Can I ask a question here?
Yeah, absolutely.
What decides the size of these tectoids?
That is a really good question. I'm going to quantify the size of these tactoids in a few slides, but I think we don't really know. And I'll just give it away right now. We have not been able to correctly simulate these tactoids. So I'd be interested if anybody knows or has any hints on how to limit the size. So in our Cytosim simulations, we have not been able to recreate tactoids.
So it is related somehow to the microtubule lengths, or it is not?
Yes, I'll show you in a second.
Okay.
It is. Okay. Just to be very briefly on these crosslinkers. And this is also another hint about why they're not merging. If you look at if we do a fluorescence recovery after photobleaching, which is a way to get at internal dynamics in a system, right? So what you do is you have fluorescently labeled objects, the crosslinkers and the microtubules, and then you purposely turn off the lights in part of them. If there's dynamics inside of the system, meaning turnover within the system, then what you'll see is recovery of that fluorescence due to the dynamics, right? Because you've now turned off the lights, but only on a portion of them. And so the ones where the lights are still on, if they're able to move into that area, will do that. And what you see is that when we photobleach the microtubules, they don't really recover. And so if you just do a kind of even bad analysis of this, where you look at this whole circle, you can see that it's not really recovering. And in fact, what most of this recovery is from is the background. The microtubules aren't really recovering. Whereas if you do the same thing on the MAP65, it very quickly recovers. The microtubules are still there, right? Because the MAP65 only binds where there's microtubules. And the MAP65 is a very labile transient binder. And so it actually recovers back into that area but the microtubules themselves don't. So these microtubules are not liquidy inside of these tactoids. And so this is kind of interesting. They're not liquid crystals, even though they have that shape. So I keep using this word tactoids. Not everybody is familiar with it. So I just wanted to give you the Wikipedia definition, or you can look it up while you're sitting on your computer. But they are liquid crystal microdomains that condense or nucleate out of isotropic phases. So the liquid crystals, they're long molecules, longer than they are wide. If they're in an isotropic phase, they're pointing in all the different directions. And then if they start pointing in the same direction locally, you'll end up with a either spherical or spindle-shaped droplet. And you can tell that they're all the molecules inside, or the liquid crystal elements inside, are all pointing in the same direction, both by the shape. So just a reminder of what does it mean to be spindle shaped. So I'm showing you two spindles. One is the spindle like you might have on a stair rail or on a chair leg. And the other one is the mitotic spindle. They have this name because of this kind of shape, rugby ball shape that they have. And then this birefringence, just as a reminder about birefringence. So if you look at something under cross polarizers that has a material inside where the inside constituent material is elongated, you'll often get a different index of refraction in one direction versus the other. And by looking at them under cross polarizers, that reveals that index of refraction. So just this plastic ware that I'm showing you here, it's a polystyrene plastic. When they extrude to create these plastic silverware, tableware, it actually aligns the polystyrene inside, and that or the polymers that are making up this plastic, and now you can see it as a birefringence. And so whenever you have a material where the molecules are kind of aligned within that material, you'll often get a birefringence where the light travels when it's parallel to the molecules inside. It'll travel differently than when it's perpendicular. And so that's where that birefringence comes from. And so we can look at our system and ask, you know, is there a birefringence to our system? So what we do is we put it under cross polarizers. We have a rotational stage that allows us to kind of just rotate the sample while we're looking at it. If we're looking at it with 0 degrees, we can see nothing, which, and then, but if it was completely isotropic, we would also see nothing. As we start to tilt by 15 degrees, we start to see something, 30, 45. So now we're really noticing that there's a much brighter area there, right where, and again, the cross polarizers are not moving. They're staying at cross. What we're doing is rotating the stage so that whatever these objects are now rotating within that. And we would expect there to be the highest at 45 degrees. And then as we continue to rotate past, we now see nothing again. And so this is what we would expect for something as birefringent. Again, showing us kind of what we already know, which is that the microtubules are probably aligned in this bundle. And now the microtubules are acting like the liquid crystal elements where the light can move faster in one direction than the other. All right, so getting to what we were just asked about with the shapes of these things. So we can now measure the length and the width separately, and of course take their ratio and give us an aspect ratio. What you expect with these kinds of condensates, you know, if this truly was a liquid crystal condensate, is that the length and the width, the aspect ratio, as you are adding more of the ability to condense. So in this case, it would be controlled by the crosslinker. So we'd expect you add more crosslinker and you become more spherical, right? And also the length, there should be a length dependence on that contour length. And so when we do this, we actually kept the microtubule contour lengths the same. And then we just added in more and more and more of the crosslinker. And what you see is that indeed the aspect ratio decreases. So it's becoming more and more spherical, as you would expect, going closer to 1. The lengths are actually decreasing. So if we look at these separately, we look at histograms of just the length and just the width. The lengths are actually decreasing, but the weird part is that the widths are not changing. So these tactoids really seem to have selected a width, and they all have that width, and it doesn't seem to matter a lot of the other parameters that we change. The widths actually stay constant, and that means that we're not really able to kind of get to that liquid crystal regime. And I should say, this is not something that you would see with a real liquid crystal. You would actually see that the width would increase, the lengths would decrease, and then that would cause the aspect ratio to approach 1. And we're not seeing that. We have some ideas about this constant width and why it would be happening. There's some ideas that if you add a rotational element, so maybe the microtubules are actually kind of rotating in the tactoid, so they're not going straight through, but they have a twist. And if they have that twist, then that would limit you. It would also cause it so that 2 tactoids wouldn't be able to merge, because the microtubules inside would be incommensurate. These ones would be twisting that way. These ones would twist this way, and that would limit your growth. So maybe some sort of geometric frustration that we're seeing here. But that's just an idea. We haven't actually had the chance to look at these at higher resolution like electron microscopy yet.
Could it be like, could it be like muscle contraction, that they are just being pulled in by, just like the myosins do? Is that the crosslinkers can do that? Simply pull them in without increasing the width?
Yes, so it's been shown with, I believe, ACE1, which is the yeast version of the same protein, that if you have 2 microtubules and you have an overlap between them with the ACE1 in between, the ACE1 will draw them together so that they become perfectly parallel. And so it's possible that MAP65 would do that as well. We haven't done that experiment. That experiment was done very, very nicely by Stefan Dietz, and where he used an optical tweezer. You could even feel what the entropic spring force was. Right. No one's done it with MAP65 that I've seen, but I don't have any good reason to think MAP65 would work very differently than ACE1.
But he had a mixture of active and passive.
He did, but I thought when he did passive alone, it still sucked in. Is that true?
I don't recall fully, so I must.
Okay.
Yeah.
But we should look back at it, and then we'll see.
Okay. Sure. Please continue.
Okay, so I'm gonna actually move on to the second story. There's more to this work, of course, and we have another paper that, like I said, is under review now. But I'll just give you some takeaway messages. So if you have no crosslinkers, you have these spindles, and we're able to recreate this using our simulations. If you add in crosslinkers, we end up with these spindles, but the shape is right. So you don't need any kind of special activity to get the shape. You really just need the microtubules like acting as long, liquid crystal type elements, that they're longer than they are wide, and then condensing those elements, right? Once you do that with a specific crosslinker, it's not enough just to use depletion interactions, then you'll actually get this kind of shape. But these things that we've created with this crosslinker, these little tactoids, they're not liquidy. So they're not really yet like a spindle in some of their characteristics. They've got the right shape, but they don't have that fluidity that we would like. And so what we're working on now is adding in activity, so we can add dynamic instability to the microtubules. We can purposely add in polymerizers and depolymerizers to really control that dynamic instability. And we're also adding motor proteins. So we're adding in these cross-linked kinesin motors in order to try to fluidize the inside of these structures so that they're not quite so jammed like they are now. Okay, so then the second tale I wanted to tell you is completely different. So no microtubules. This is a complete departure for my lab. We started this work a couple years ago, and I'm really, really excited about it. So I'm gonna try to orient you again about this step. So normally, if I was there in person, what we would do is a demonstration. I would pull out a bunch of balloons and I'd throw them into the audience, and you would play the game called Keep It Up. So I don't know if any of you remember what Keep It Up is, or if you've ever played this. So this is a game for lonely children. Possibly you become a physicist when you grow up. You don't need a partner to play this game. You basically have a single balloon and you try to keep it up, right? So it's that kind of a game. Now I have a movie. This is a kind of a new twist on Keep It Up where they've added pool noodles. But it basically, I guess this was a news article about how to keep your children entertained during the pandemic. I'm not really sure, but this is one way if you're worried about keeping your children entertained. You give them a single balloon in their hands or a pool noodle, and then they can play Keep It Up. Now, when you watch Keep It Up, so let me play that again. my question for you, and this is for the chat, so we're going to do another kind of quiz thing. So I have a couple of questions for you. Is this process stochastic? So you're going to watch these kids play, keep it up. Vote in the chat if you think this process is stochastic. What do you think? No, I got a no. I'm going to play it again. Okay, so maybe some of these children are very coordinated. Let's watch this first kid. Is that stochastic? Watch the kid with the yellow balloon. Don't watch the kid with the red balloon. She's too coordinated. Go back over here. Kid with the yellow balloon. Here we go. Watch where the balloon goes. Is she? These kids back here are also pretty random, it seems. Right. So I do think it's pretty random. Obviously, this child in the front, not my child, because way too coordinated. But if we were to do this in person, where I threw a bunch of balloons out to you, any one person could choose a direction, right? But then each person would choose a different direction, and the balloon would end up migrating throughout the lecture hall, and it would actually migrate in a way that looks like a random walk, because each person's kick would be, it's literally a run and tumble, right? So each kick basically chooses a different direction, and you're giving it a certain distance that it'll kick to the next person. And so you end up with something that's random, like stochastic. Now, let me ask you another question. If we were to do this process, is it equilibrium? Is it in equilibrium? Go ahead and vote in the chat. I got one vote. Nobody's gonna vote? No, it is in equilibrium. That's right. It's not equilibrium. It's obviously not equilibrium. It's getting kicks. It's not, they're not random. They're random in direction, but they have activity, right? The person is pushing. And if we were doing this again together, you would be pushing it. You'd be using your arm muscle, your actin in your muscles and all this stuff, right? So, right, it's not equilibrium. Okay, so a lot of these systems in biology are stochastic. We talked about that from the very first movie, fluctuations, right? They look random. And so, but you can get work out of these systems. Now, we already know that we can get work out of a random system, right? We know from thermodynamics, there's a mechanical equivalent of heat. And we can just have an ideal gas. There's not something that's a lot more random than an ideal gas, right? Things moving in all directions. If we apply and put it under pressure and lock in our piston with a pin, the gas itself, just the kinetic energy of the gas will try to make it push up, right? If we were to unlock the pin, then the piston would move up and we'd get work out of our system. All right, so we know that we can take a random system and get work out of it. In that case, we had to give it a lot of work. What if instead all the things are active, right? So that's what we're going to talk about today. Uh-oh, what did I just do? I don't know. Okay, so the other thing we learn about in thermodynamics is Maxwell's demon. And so Maxwell's demon, the idea is that you have a hot box and a cold box, and then there's a demon sitting there opening the door between, so that every time a hot molecule moves from the cold, is coming towards from the cold side, it allows it to go to the hot side. And every time a cold molecule is in the hot side, it allows it to go back to the cold side, which would allow you to make the hot side hotter and the cold side colder. And when we think about this as a thought experiment in class, we realize that, we think, this is funny, this is not possible, right? You would never put a system together and have a hot side getting hotter and a cold side getting colder, right? That's not how entropy works. That's not how the second law tells us things go. And it's true if you think about it like just as an ideal gas only. But then if you realize that I'm sitting in an auditorium, or you're sitting right now in your rooms, I don't know where you are, and you are in fact a hot bath that remains hot in a cold surrounding. And so in fact, the idea of the Maxwell's demon is not, it's not something to laugh at. It's not just a thought experiment, it's real. And Maxwell's demons exist in biology all the time, and you are currently full of them, which is what's keeping you warm on the inside, even though the outside temperature of your room might be cooler than you. And so we have these are real in biology. And in fact, no place are they more obvious in biology than in ion channels. So if you have a membrane and you have an ion channel, that ion channel can be a selective ion channel that chooses what type of ion is allowed to go through it. It is literally a Maxwell's demon. And a lot of times those ion channels can be even passive, but they can also be active. And many of the reasons why we're able to maintain our temperature is this active, this non-equilibrium nature. So the Maxwell's demon, it's actually fine for you to make a hot bath hotter and a cold bath colder, as long as Maxwell's demon himself, the little demon, is using energy to do it. And so that's what happens a lot in biology. So in biology, there's tons of noisy random processes. So for instance, on the left, I'm showing you bacterial swarm. On the right, I'm showing you some Janus particles, which is a synthetic version of kind of what you're seeing on the left. And in both of these cases, these things move around, but there's a random element to their motion. And in both of these cases, even though these processes are random and noisy, they're obviously using energy, and that means we can rectify them. And so what I'm showing you on the left is a rotor where the bacteria, the same type of bacteria, are now being captured, and they are pushing the rotor around. So the fluorescent things that you're seeing when this moved into the fluorescent mode are the bacteria. You could even see some individual bacteria moving in the background. And now you can use these bacteria. And again, what's happening is you're rectifying the motion over here. On the right-hand side, what you're seeing are Janus particles. And again, these Janus particles in this case actually are magnetic, and they're able to come together and they also form these little spinning pinwheels, which you could get work out of. And so in both of these cases, these little active particles, this active matter system, has a little tiny onboard motor, right? In the case of the bacteria, it's the flagella, which you can actually see fluctuating here and rotating, which is pushing it forward. In the case of the Janus particle, it's a chemical gradient that's being created in these particles. And now both of them can now be rectified, and you can get work out of a noisy non-thermal system. All right. So biology does this all the time. So I probably don't have to tell any of you this, but I'm just giving you 3 examples of where we are able to use this work. And many times this work is happening on a molecular scale, so on the nanoscale. And yet you can actually still get micron scale or higher macro scale responses. So this Touch Me Not plant is actually one where when you touch it, it opens a mechanical sensitive ion channel that creates an ion gradient that causes the leaves to close up. So that's what you're seeing there. I showed you this movie already of mitosis. Obviously the microtubules are pushing and pulling these chromosomes, and then ultimately helping to split the cell in 2. And then finally, this wound healing assay, this is actually in a microscope in the dish where you've grown a confluent layer of cells. You can scrape across the cells, and then you can watch them as they grow back in. And so all of these processes are due to that activity, but they're all a form of rectifying the noise into work. And so I think this is a grand challenge of learning from biology so that we can use these to create new materials that have really cool properties. All right, so I showed you this earlier, these Janus particles. I just want to remind you that this is a synthetic active particle. This is probably the gold standard for active colloids. The idea is that you have a particle that's coated on half of its surface with a metal, and that metal, when you put it into a chemical bath and shine a light or whatever it is, it performs a chemical reaction. Some push, some even create bubbles, and so they're like literally you can see little bubbles coming out the back, and some pull in the gradient. But either way, the results is that this particle will propel in a unidirectional motion, and that Janus, or that two-faced nature of the particle, is what gives you, right, you need to break the symmetry. So that's how the symmetry has been broken on this round particle in order to cause the propulsion. There's been some really nice work from a couple of groups where instead of using the metal, you can actually coat this with enzymes. And these enzymes, when performing their chemical reaction, again, they're changing the chemistry locally, just like the metal does, and that can also result in the propulsion. Now, that begs the question, is it possible for the individual enzyme to also be a propulsive unit of itself? And so the big differences here, obviously the Janus particle is really big. It has slow rotational diffusion. You have many of the propulsive units, the propulsive enzymes on it. It has both slow rotational and slow translational motion, so that when you have enough of the enzyme on it, you get faster propulsion. For those of you who think about this stuff, the Pecquelet number, which is the ratio between the kind of directed velocity motion and the rotation or the diffusional motion, that ratio is greater than 1, which means that you get kind of a directed transport, and it'll change over time. And that's what we saw with those Janus particles earlier. They kind of had more directional motion and then less rotational motion. Now, if we think about the enzyme is at a very different size scale. Down at 4 nanometers, it has an incredibly fast rotational motion. It also has a fast translational motion. And even if it is able to propel, it's going to propel itself a lot slower because there's only one of it. So it's performing its chemical reaction. If it's able to propel, the Pecquelet number is going to be less than 1. And what you would expect to observe is fast diffusion. So if this enzyme is an active unit all by itself, we would still expect to see a diffusive type of motion, random walk, Brownian motion, but it should be increased if there is some sort of propulsion going on. In a sense, you can think of it as a random walk where you've increased the step size because it's able to propel every time before it rotates. The other reason why we're interested in this is that there's been several studies dating back for now 6-7 years, where people have been looking at the inside of cells and seeing that those inside of the cells is jammed. It's chock full of proteins and polymers, and in particular inside of a bacterial cell, there's this large DNA polymer that's plasmid DNA and chromosomal DNA, and it's just really everything is really stuck in there. And very large objects, if you turn off all enzymatic activity, those objects actually become jammed. They cannot move. Very small objects like a single GFP will still be able to move around. But as you increase the size of those objects, it's very difficult to move inside of there. And so it's been hypothesized that there's like a secondary effect of having all these chemical reactions and all these enzymes inside of cells, which is to loosen up the interior. In a sense, the enzyme acts like a temperature gradient, an activity gradient. But it's non-equilibrium version of it. So it helps you to get the mixing inside of the cells. So just to be clear, I'm going to use the word enzyme, which is going to be it's actually a catalyst. So the enzyme itself is not changed during the chemical reaction. It's just causing another chemical reaction to occur. That chemical reaction, chemists often call the stuff that comes in the substrate, and then what it changes to the product. We can call it the fuel and the exhaust for this, our simple language here. As physicists, it doesn't really matter, but the point is we're going to have chemicals that go in and they come out changed, and the enzyme will stay the same. The enzyme we're going to use is urease, and it takes urea and and water and turns it into carbon dioxide and ammonia. And I think that this equation is messed up, so I apologize for that. I meant to update this equation because I don't think if you do the math, I don't think it works out. This work has been done by Mengzi, who's a grad student. And again, she started with me at UMass and has moved to Syracuse. She has a second story on this stuff as well, but I'm going to tell you about her first paper, which was published in PRL. This idea that enzymes can move faster has been around for a little while. In particular, Ayusman Sen has been looking at this for quite a while. He's looked at a bunch of different enzymes. One of them is catalase, one is urease. He's had a few papers. These are from 2 of his papers where he could see that, yes, the diffusion of these enzymes was higher as you add more and more of the substrate. And so you might say, well, that's great, but actually it's not very big. It's not a very big shift that you're seeing here. And which is fine. That's not a big deal. It's still statistically significant. The problem is that when people were doing these experiments, and these experiments especially, everybody was using the same technique. They were all using fluorescence correlation spectroscopy. So in fluorescence correlation spectroscopy, you have a molecule, and you have molecules, and they're diffusing. And then you have a diffraction limited spot, and they diffuse through the diffraction limited spot. And when they do that, they're fluorescent molecules, so they fluoresce. And then you get fluctuations in the fluorescence, and you are interpreting those fluctuations in the fluorescence as all being directly due to the mobility or the motion of the object as it's going through that light cone. Now, the problem with that is that fluorescence can fluctuate anyways, even if it's not moving. And so you could end up with artifacts that are not actually just due to the molecule motion. So it's even less direct and removed from a regular scattering technique. But you can't do a scattering technique very well with these because they're very small. So we're on the nanoscale. And so we know that you need to scatter around the same size wavelength, right? And so, so people have now done scattering techniques. And so since we did this work, lots of people have been able to recapitulate this with multiple techniques. But at the time that we started working on this, FCS literally was the only thing that people were using. So you look at the correlation function and you see the decay. And what happened is in 2018, somebody said, no, FCS has a lot of artifacts. And they showed a variety of different ways where you could see kind of fluctuations. And in particular, one example is quenching or other quantum effects that can occur. And so by basically for the enzyme, just having the ability to bind and release product actually changes the environment for the fluorophore, and that can cause you to have different fluctuations in the fluorescence. And so they said, okay, you know what, all this is artifact, and you know, it's all been done by the same technique and it's all artifact. And so we decided that we wanted to actually do a different technique, and we wanted to directly image the molecules as they diffuse. The problem is these molecules diffuse really fast. So what we did is we slowed down that process. We did that by adding methacellulose, again, our favorite kind of big fluffy polymer, to help slow things down. And then we also put a PEG brush on the surface, so that we have a polymer brush on the surface. We're going to use our turf imaging, so we're only going to look really close to the surface. So these molecules have to be near the PEG brush in order to diffuse. And we're kind of lucked out that this PEG brush actually does affect the diffusion locally on the surface. And it slows it down by about a factor of 300, which is about what we need in order to see this stuff with our microscope. So we're very lucky this process has worked. And we've done some more analysis to try to figure out what the PEG brush is doing. And it really is just seems like it's reducing the diffusion coefficient. When Mungzi first started this, and these are some of her very first movies, so it's really hard to see because we had a green molecule and it turns out the whole world is green, has green background, but you can actually track this green molecule. So here's the track coming up. And so we can take that track, which in the XY over time, and we use that trajectory to get a mean squared displacement. So this single molecule gives us one diffusion coefficient. And now this is the power of single molecule imaging. Every single particle can give us one diffusion coefficient, and we can create a histogram of diffusion coefficients. Instead of like with FCS or with scattering techniques, you get one number out and it's already an average over everything. That means if you have some that are fast movers and slow movers, you're never going to see the difference between them. In this case, we can actually track every single molecule and get the distribution of the diffusion coefficients. With the mean squared displacement, I want to point out that we would expect for diffusion for it to be linear, and that linearity, the slope should be give us the diffusion coefficient. We are assuming that we're in a two-dimensional diffusive state, although obviously it could diffuse in 3 dimensions. We can only see 2-dimensional diffusion in our microscope. So we're fitting it with 4D times the lag time. Now we can do this with and without the urea. So that's the experiment. The experiment is what happens when you add the fuel or not the fuel. So this is urease. Remember, it takes urea and turns it into these other products. When you have no urea, you can see these individuals diffusing. And when you add the urea, it really becomes much faster. The playback on these is the same. It's 1 second per second. The number of frames is slightly different. We had to increase the number of frames when we first started doing this. This is one of her early movies, not as early as the last movie you saw, because we changed the fluorophore to get better signal to noise. So this is a Alexa 647, which has a better signal to noise for better tracking. And but the first time that she did this, she did all the control experiments first, right? This is what you do when you're a good scientist. You do your control experiments first. She said, okay, I can track it. It's all good. I know how to take this data. And then she added the molecules with the urea, and she came to my office and said, Jenny, all the molecules disappeared. I was like, what do you mean they disappeared? And she goes, I can't see them anymore. And when I came in, she was right. There were no molecules there anymore. I go, well, did you pipette them in? Right. That's always the first question. Did you actually add the molecules? And she did. And what it was is that it was moving so fast that it was now too fast for this frame rate. And so we had to increase the laser power so that we could get more photons in, so that we could take faster images. And then when she took the faster images, she was able to see them again. So again, this is kind of like pretty early experiment of what she does. She now obviously takes everything at the same time frames between both, but for our first couple of ones, she had to take this. She actually had to change the frame rate in order to see it. If you now track each of these and you do the mean square displacement, you can see that the one with the urea in blue is moving much faster. Obviously, the slope is much higher than the one with the buffer only. And you might say that we just picked two lucky examples, but again, the beauty of single molecule imaging is that we get this whole histogram. And so now what you see is that there's a significant shift in the data when you have the fuel present for that enzyme. And it really does shift towards faster diffusion coefficients. We obviously still have some that are slow and some that are fast, but we also don't see splitting. We don't see some that are, we don't see two humps or anything like that. But that is something that would be given to us here. We can do some other control experiments. So again, because we're using this polymer brush and the methylcellulose to help us slow down, we wanted to make sure that the diffusion still scaled with size. So we can track single GFP, green fluorescent protein, or we can track labeled catalase, which is just a different enzyme with no activity present. And we see that the diffusion does in fact scale with the size, as you would expect. Mengzi has now done other experiments looking at size and viscosity. Everything seems to track the way that you would expect. We can also make sure that urea is not some sort of miracle slippy slidey molecule, that anti-viscosity molecule. We can do that with GFP. So we do GFP with buffer or GFP in the presence of the urea. And we see actually the urea, if anything, it causes it to be a little slower, not a little faster. And so all the enhancement that we're seeing, we are chalking up to the enzyme activity. We can do this for a bunch of different urea concentrations. And again, now you can see that the histograms are all shifting. And then we can plot it versus urea concentration. And much like you would expect for an enzyme, you can actually see that it does depend on the urea as an inverse hyperbola, which is exactly what you would expect for enzyme kinetics. And so we're very happy not only that it was moving. The other thing that I want to point out is that our shift in diffusion is much higher than what people had seen previously with the urease. And so they had seen maybe a 20 or 30% increase in diffusion. We're seeing a factor of 2. So that was kind of also exciting for us to see, and we're imaging this directly. When we were doing this, there was some theory coming out, in particular from people who've been looking at active matter and really now thinking like, how could these enzymes be doing this? And a couple of the theories were saying, well, this is a collective effect. And what's happening is these enzymes are actually changing their environment. And in particular, there was one study that said, or one theory that said that they thought they were changing the environment by increasing the temperature of the environment. And they said, look, if you add more and more and more of this enzyme, then you're going to increase the temperature globally, and that's what will cause this increased diffusion. So it's not that you actually have some special propulsion, it's really just that you've increased T, and in the Stokes-Einstein equation, if you increase T, you're going to increase D. And so there was another study saying, okay, the same thing happens, but it's viscosity. You're changing the viscosity, and now you can change it globally. So for both of those studies, they made a prediction that said that this effect should depend on the concentration of enzyme. So all of the experiments that you've seen so far were done at very, very low enzyme concentration at 92 picomolar, where all of the molecules in there were labeled. So what we decided to do was keep the amount of labeled molecules the same, and then just add in a high amount of unlabeled molecules in the background. This would directly test these models that say that it's a collective effect due to the fact that you have many molecules that are doing this. So we have 40 nanomolar unlabeled urease or enzyme in the presence of the same 92 picomolar. And so we tested this with both of these. And what we find is that there's no collective effect. So at least at these concentrations, we are not globally changing the temperature. We are not globally changing the viscosity. This is truly a single molecule, single enzyme level activity. So whatever this is, whatever it's doing, however it's actually working, which I should say is still unknown and needs good theory that we can test, it's happening at the single enzyme level. So we're very happy that there were theories out there, but those theories don't work. So now we need new theories. There was another theory brought to us, this one at March meeting when we presented. Somebody said, well, one of the things is that these enzymes are multimers, right? So the catalase is a tetramer, the urease is maybe a trimer or a hexamer. You've got multiple ureases that come together into a complex. And so they thought, well, maybe what's happening is when you perform your reaction, you're actually falling apart. And when you're smaller, you can diffuse faster. And so we wanted to test that. And what's great about single molecule is that this is definitely something we can test. So what we do is we do single molecule photobleaching. We now, instead of having the polymer brush to keep the enzyme off the surface, we actually just purposely stick the enzyme to the glass. We've saturated it either with the urea, the fuel, or without the urea. And then we just stick them to the glass in that state. It freezes them in that state. And now we can add the light and cause it to purposely photobleach over time. What you see is for any one enzyme, you see a little spot, and you'll see that the intensity will decrease over time as it photobleaches. If we get the intensity just right, we can actually see individual step down of that photobleaching, which will allow us to count the number of fluorescent molecules. We also have to make sure that our enzymes, we know their labeling ratio. So we made sure that we were labeling at around 1 fluorescent molecule per enzyme monomer, so that if you had a trimer, you should expect to see 3. Now that's also statistical, so there could be that some have 2 and some have 1, but on average, each single monomer should have a single fluorophore. So now we can use this process.
Professor Ross,
yeah.
Sorry, so 1 hour is already over. So can you wrap it up soon?
Yes, I definitely can. Okay, I'm almost done, I swear. So so this is just showing you we can now look at the distribution of the intensity steps, and you can see that the 0 urea and 1 millimolar urea have the same distribution. And so we actually see no difference in the oligomerization. All right, so this is the end. So I'm sorry I'm going over, but we see is that the single enzymes do diffuse faster, which implies that they're active elements on their own. That activity really depends on their enzyme activity. It's not a collective effect, and it doesn't seem to depend on their oligomerization state, or at least their oligomerization state is not affected by their fuel, as you might expect. This paper was published in 2019, just last year, in PRL. We are looking for other theories. So if you have theories that you think we could test, please send me an email. I'd be happy to chat with you, or we can chat right after this is done. There's a lot of open questions for this. So we don't know the fundamental physics, right? Stokes-Einstein says that the diffusion doesn't have anything to do with enzymatic activity. It only depends on the size and the viscosity and the temperature. And now we're saying that enzymatic activity is another thing. Is it like temperature? We don't know. So I think there's a lot of fundamental physics that we don't really understand about this process. The other thing is that you could use these enzymes as active matter. So we have a new NSF grant where we're designing DNA origami. We're sticking the enzymes on, and we're trying to create mesoscale active matter to try to understand how the size scale of the active particle will affect the activity. And then of course, can you get this internal mixing, right? So can you mix nearby? Is this actually something that cells are using to keep themselves active inside and keep everything fluidized inside? All right, so I just want to point out that these aren't the only projects we have in the lab. If you're interested in what we're doing, we have work on microtubule severing enzymes, motor proteins, and we're also doing composite networks between actin and microtubules. So there's a bunch of things going on in our lab, and it's really exciting. We moved about a year ago, and things are actually functional now. So it is a really exciting time. So I want to say thank you so much for listening. I apologize again for going over. Also, thanks to the funding agencies and, of course, the people who really do the work in the lab. This is a few of my labbies, as I call them, pictured in this movie trailer for their kinesin preps that they've been making lately. So I appreciate your time and attention, and I'd be happy to take any more questions.
Okay, so can I start?
Sure.
Okay, so this is Debashish. So very nice talk and a very interesting, experiment puzzles. So let me make a speculation, okay, because you allowed audience to make that last part. So when you use the Stokes-Einstein relation, so there, of course, you have the radius or diameter of the particle. Now, is it possible that during enzymatic activity, it actually becomes more compact? So that way, this effective viscosity goes down. And so by Einstein relation, then of course the diffusion constant would become higher. Did you see the conformational change when it becomes more compact?
No, we haven't seen that. That's definitely a possibility. So actually, that could also be done with single molecule imaging, but we would have to, I think we could do it with FRET, right? So if we had two monomers labeled with different colors and they came together in one, we might see that if it becomes more compact or even more elongated, right? If it just changes shape, that could result in a change in the diffusion constant. I agree. That has never been done to my knowledge, but it's definitely, I think, so it's a little bit difficult to get the fluorophores in locations where you exactly know where they are. But that you would have to really engineer and design it. But that is definitely a possibility that we haven't yet tested and no one's tested yet.
And another question, regarding the first part of your talk. So if I recall correctly, Nedlich had in his simulations also seen, of course, maybe because he had motors, I don't know, something which was asters. You don't have any asters. You have only spindle-like structures. So is the aster there appearing only because of motors which you don't have? Is that possible?
Yes, I think that's right. And so what happens is he had these cross-linking motors. And they would walk to the plus ends and kind of gather all the plus ends together. And people have now done this also with dining, and you can, you've been able to do it in experiments. You can do it in the simulation, and we do not have motors yet. So that's one possibility of what could happen if we add the motors to our system. In particular, if we add the motors and reduce the cross-linkers, we might actually see asters instead of the tactoids. We're still working on getting the motors. We're actually trying to use these motors that have a light inducible so that we can actually turn it off, create the tactoids, and then use light to turn the motors on locally, just where we want to see the activity. So, because I'm worried that if we have a global activity, we'll end up getting what Zvonimir Dojic gets, which are like these big liquid crystal kind of networks of microtubules, and that's not necessarily what we're trying to see.
Okay. Thank you.
Any other questions or comments?
So let me stop the share.
Yeah. Anybody else? Okay, I don't see any more questions or comments. So in that case, let's thank Professor Jennifer Ross again for a very nice, clear, and very exciting talk. Thank you very much.
Thank you very
No
much
problem.
for a very, very nice talk. Thoroughly enjoyed that talk. Thank you.
Great. Thank you.